Hey guys, welcome to Subu Medicine, and this is Neurology. And today we'll be discussing about the development of nervous systems. And I've got to realize and also got to know from several people that this is one of the difficult concept to understand. So I'll try my best to explain you in a very easier language, so that way you can have a greater understanding of this topic. Okay, so let's begin. So before even we go and talk about how the neurulation takes place, we have to start from beginning, like what is happening in the first week. And I'm not going to go super detailed about that, but I'm just going to quickly recall that information. Okay, so first week, you guys know that. Okay, first week is what? Fertilization takes place, right? Let me write down here. Fertilizations. And then, let's say at the end of the week, there's going to be a implantations, right? So this is what is happening. Around like day 6 or so, the implantations occur, right? So when implantation occurs, usually the cells appear to be in the structures. So if you see right here, see, this layer, we call them a inner mass. And then this outer layer, we call them outer mass. Because these guys right here, this inner mass is the one that's going to, our inner mass is also called, you can simply call this a embryoblast. And this one is called trophoblast. This outer mass is called trophoblast. And so this is what, and this is called like blastocyst cavity, all right? Because this inner mass, embryoblast, this is what's going to form your embryo, right? So, this is usually, this is happening around like day 6 or so, right? Day 6, day 7. This is the phase when the implantations occurs, right? Then after that, what happens is like around the day 8 or so, these cells of these trophoblast cells, they start differentiating. Okay, they start dividing into 2 layers of the cells, the outer cells, and these outer cells you see in the pink color, we call them a syncytial. Okay, let's just write here. Syncytial trophoblast, because this is really important, because these are the multinucleated cells and very, very important for like secreting HCG hormone, human chorionic gonadotropin hormone. Because if you get to see this in urine, then we usually say that this is the indicative of pregnancy. All right, and then this layer right here, all right, this guy is right here in the black. What is this called? This is called cytotrophoblast. It's called cytotrophoblast. Because remember, the week 2, all the layer, everything happens, like, we also call them everything divides into 2. So basically means that this trophoblast get divided into 2 parts, which is called syncytial trophoblast and also cytotrophoblast. That happens. And not only that, this embryoblast right here, this form, they also, what has happened? They form something called bilaminar disc. And how do they form the bilaminar disc? If you come here, right, look at this structure right here, right? This layer right here on the upper layer, because they differentiated into 2 layers of the cells. And the first layer, they appear to be a columnar in nature. So these columnar cells, we call them a epiblast. This is called epiblast. And then this green in color right here, okay, let's just write here. This green in color, this green color right here, right? These green cells are called hypoblast. All right? And then, look, there's also, from here, the cells from this hypoblast started migrating and make layers, okay? Layers, and these are called like extracellular membrane and all. Okay, but I'm not going to too much focus on that. But just know that this embryoblast will differentiate into 2 layers of the cells. One is called epiblast and the other one is called hypoblast, right? And the hypoblast is cuboidal in nature. And this cavity right here, we'll call them a primitive. It's called primitive yolk sac. And then this guy, this epiblast, makes this layer. What is this called? This is called amniotic membrane. And this right here is called amniotic cavity. All right. So this is an epiblast and this is a hypoblast. That is what's happening. Okay. In your second week of your development. And also one more thing that also happens that you should get to know here is that, in the second week of development, what happens is that, some of these cells from the hypoblast, what they do? They start migrating. They started migrating upwards, and they sort of, they were cuboidal. They became columnar, and what happens is they fuse with this epiblast cells, which are already columnar. And when they fuse with this, they make like thickening or circular structures. And that circular structure is right here. Okay, let's make this out. And this circular structure is called prochordal plate. All right, and it's really important. And what is the importance of this prochordal plate is that this prochordal plate helps us to distinguish cranial and then caudal axis. Let's say cranial and then caudal axis. What does this mean? Caudal axis, which basically means is that based on the locations of this prochordal plate, we can able to say that which one is going to give rise to the formations of your head and neck, and which one is going to form your anus regions or your lower extremities based on this prochordal plate. All right, so that's the importance of this. And not only that, there's also, you see like some of the layers of thickening, you get to see in front of this prochordal plate, and that is a membrane, which we call them a buccopharyngeal membrane, because this buccopharyngeal membrane, that's what is going to become your future mouth. Okay, and then also there are also some of the cells like down here on the back right here, which is called cloacal membrane, and this is what's going to be future anus. So you can simply say this is mine. All right, so if I right now you're looking in a more coronal view, right? If I look at this way, I can say this is my anterior view, and you can say this is a posterior. You can think about this is a cephalic end, okay? This is a caudal end. This is a anterior end, this is a posterior end, this is a talar end, all right? So this is what you got to think about. Remember, these cells, the prochordal plates are formed by the cells from the hypoblast, which migrated and sort of like changed the shape and sort of like, it started getting thickening and created this structure, which is called prochordal plate. Okay, now, so you guys know this, right? So this is called, as I said, this is a primitive yolk sac, and this is called amniotic cavity, right? And these are all happening during the second week of the development. All right. Now, not only that's what is happening here is that, okay, if I let's say if I cut this, all right, or if you want to look at from the, let's say from the top, all right, and if I just want to draw this, let's make this structure. It's going to look something like this. What is this layer? You see here. This is going to be this is going to be my epiblast right here, right? And then this is what my hypoblast, right? And this is my precaudal plate. Okay, so if I'm looking for the top, you can think about this end right here. This is my posterior end, and you can see this is an anterior end. Okay, so if this is my, let's say, anterior end, and this is posterior end, what really happens is that these cells from the epiblast, they started like, going through the process called morphogenesis. What does that mean? Which basically means the cells from the posterior end, let's say the posterior end, right? Let me make this in this color right here. Okay, let me make this. Cells from the posterior end right here, from the posterior, they started, elongating and moving upward like this, upward, right here. So when they start moving upward, elongating upward, and they're moving towards the anterior end, and they also make like knob-like structure like this. Okay, so basically, some of the cells near from the epiblast, from the posterior end, they started proliferating and migrating towards this precaudal plate, all right, or to the anterior end, and they form these structures. And this structure is called what? This structure is called, this is what is called your primitive strip. And this guy right here, this knob-like structure, that is what's called your primitive node. All right? Now, so this is what's happening down here, right here. Now, you saw this is a thickening like a structure right here, right? And then also, like, if I quickly make this, this is going to be just a structure like this. Okay? And if I quickly label this, what is going to happen is that, Okay, like quickly. And then what is going to happen is that, these cells in between right here, they started like degenerating. Like the middle of the cells start degenerating and they'll form structure like this. See, there's no, so if you say this is a posterior end here, let's say this is a posterior and this is anterior. You see there's like no any cells in between, right? This is no. So when the cells in between they degenerate, we no longer call them a primitive strip, but instead we call them a primitive groove. Primitive groove. I'm just like, trying to go a little more detail. And, there's so much these words like primitive pit, primitive node, so I'm just trying to help you to understand what this means, all right? And this right here, we no longer call them a primitive node because it creates this cavity. We call them this primitive pit right here. That's what's happening, right? So remember, as I said, the cells from the epiblast, from the tail end or from the posterior end, they started proliferating and they started like making and elongating towards this precaudal plate, okay? And that make the structure called primitive strip and the primitive node. And the cells in between, they degenerate and then they form something called primitive group and also primitive pit, okay? That is really, really important. This is really important. And now what's going to happen here is that, the cells from all this, now what is happening, this epiblast cells, started, secreting a certain type of growth factors like fibroblast growth factors. There is also a BM, let's say, bone morphogenesis protein like 4, or like N-cadherin proteins, right? Or fibroblast growth factor 8, or NCAM also. They started like, secreting. And because of that, what is going to happen here is that the cells of these cells from this epiblast, it started migrating, like let's make this as different color. Started migrating maybe like this direction right here, going this direction, okay? That's what is happening. And when they started going this direction, what is going to happen here, guys? It's a very important concept. Let's write it down here. When they started going down here, okay, some of the cells go down and then from the primitive, this group start coming down like this, okay? So when it started coming down here, what is going to happen here is that when it's coming down here like this, the cells are started, let's erase this. And I'll make it a green too, all right? The cells, I'll make this in here. So what is going to happen is the cells start coming down here like this, and it starts making like another structure like this, okay? Because cells start coming down here and you start making structure like this. And what is the structure they're forming? They're basically replacing these hypoblast cells and making another layer of the cells. And what is that called? Okay, so remember, the cells from the epiblast started migrating here, and from the primitive group, this is going like, moving from everywhere and coming downward and making another layers of cells, replacing the hypoblast. Remember, guys, it was this hypoblast layer, right? It replaces by this cell. And now these layers, we no longer call them a hypoblast. What do we call them? We call them a, very, very important, sir. We call them a endoderm. That's what we call them. Okay? And not only that, they'll continue coming down, and they start making another layer of cells right here. Okay? And this another layer of cells they make, another layer, we call them a mesoderm. And then there's still, and the rest of the cells that are still, that are here, this is no longer called them epiblast, we call them a ectoderm. Okay, let's make this as a ectoderm. So now, what is this process called? Whenever there's from the two layers of cells, the bilaminar disc gets converted into this trilaminar disc. What is that process called? This process is called gastrulations. Let me write it down here. What do we call them? I call them gastrulations. And gastrulation is basically when you get to see 3 layers of this layers of cells, which is called ectoderm, mesoderms, and endoderm. Remember, guys, the formations of, whenever we see this primitive streak formations, usually that is indicative of that, the trilaminar disc or trilaminar disc cells formations has started. That's what this primitive streak tells us. All right. Now, after this, now basically look. Now we have these 3 layers of the cells that has formed here. Right. Now, there is also one more thing that happens. From some of the cells right here, they move. They come down here to this primitive pit right here. Okay, and then they, instead of going downwards, they started going. Let's make it a different color. They started limiting it black. They started going like this, moving upward like that. And they make like this another structures. Like this, like tube-like structures. What is that structure called that making tube-like structure? Because they're moving, like coming down the primitive here, and they're moving laterally. Okay? Laterally and upward they're going towards this precaudal plate. This is what's called your notochord. All right, it's a tube-like structure, and that is coming from the cells near the epiblast that is going to this primitive pit and kind of like moving upwards, upwards, and they're making this tube-like structure, which is called the notochord. All right, now let's talk about this. All right, so if you see, like, we don't want to forget, this is going to be a precaudal plate, and you're going to see here that's going to be your buccopharyngeal membrane, and here down here there's going to be your, cloacal member that's going to give rise to the anal in the future. And then, so this is going to be ectoderm, this is going to be mesoderms, and this is the green is my endoderm. We created this, right? We created this now. And look at what is this red color. So basically what is happening here is that usually if I want to make something, it should look something like this. Let's just make this as, these are tube-like structures because this notochord I made it in a circle, but let me use the red again. So basically I can say it's going to be a tube-like structure like this. Notochord. This is going to be your notochord here, okay? It looks something like this. Right here. And there's the importance of this notochord. So what is going to happen here is that, when the notochord is formed, right? This notochord, what they do? They notochord started inducing some growth factors. Like, for example, I already mentioned this, like this, fibroblastic growth factors, or like noggin proteins. I'm going to write down noggin. Okay, this is an inductor protein. All right, noggin, or like, fibroblast growth factor. Number 8, bone morphogenesis protein 4, okay? So basically, this notochord will start secreting like different types of this growth factors. And under the influence of this notochord, what is going to happen? There will be formations of something called neural plate. And how does it look like? You see, look at here. Look, come here. Look at this. There's a formations of the thickening of the structure that you get to see. Okay, the epiblast cells started migrating and it's creating the thickening in the midline. Okay, and that is what's called your neural plate. So if you see this right here, this is what is called neural plate. So basically, in exam questions, sometimes they ask this, like, what is the goal of the notochord? Notochord induces the process of neurulation. Notochord induces the process of forming your central nervous system. Is very, very important, okay? Because in adult, what happens with the notochord is that, in adult, this becomes a remnant. It becomes a remnant. Okay, and what do we call the remnant? We call them a nucleus pulposus. And what do we find that? Okay, if let's say this is my intervertical disc. One here, okay, and there's another one here. Let's say these are 2 vertical discs. In between these 2 vertical discs, these are called, there is going to be the gel-like structure, which is very, very soft. This one right here. And then on the lateral side, you're going to see a ring right here. This ring is called annulus fibrosus ring. Okay? This is called annulus fibrosus. And in between within the annulus fibrosus, you see this structure right here, okay? That is what is called nucleus pulposus. And this nucleus pulposus is a remnant of your notochord. Okay? So this is also something that sometimes they can ask you. All right? Now, so notochord induces the formations of something called neural plate. And this is very important because your neural plate is it's like a flattened type of structures like this, and you get to see in right here, what is this called? An ectoderm right here, right? Because this notochord, we can simply it get differentiated into neuroectoderm to form neural plate. As simple as that. Okay, so this notochord again induces to differentiate into neuroectoderm, because this is going to be neuroectoderm right here, to form a neural plate. That is something that you remember. And what is going to happen is that this neural plate, and this, remember guys, this is taking place like, after third week, right? Around like day 21 or 22, the neural plate gets formed, all right? So now, this neural plate, what is going to happen is that the neural plate is going to go something called folding process. Again, right now you're still looking more in coronal view, right? So looking from the top. And when neural folding happens, what is going to happen here? That's something you remember. When it starts folding, okay, it creates this structure, which is called this right here, the line we call the neural groove. Okay, you got this point? Okay, let me explain you again. Okay, let me use something. Look. All right, you see this story? You see this hat? Okay, this is a national hat of Nepal. So you can imagine this as, let's say, you can simply say this is a neural plate right here, okay? You can simply say this is a neural plate right here. So what is going to happen here is that it's a flattened structure, okay? So what is happening from the midline, the cells are started coming together and start like folding like this, all right? When it started folding from the midline, it started folding. So let's just make this. Started folding like this from the midline right here. Like folding, you see? So when it folds from the midline, right? Look, what came out? If you see this, right, what came out here right here? Because these guys later, they get separate out, okay? This gets separate out. And that, when this gets separate out right here, this structure right here, we call them a neural tube. Okay, and when it started folding in the middle right here, look, it started zipping upwards and also downward. That's what is going to happen. First, it started go upward and start fusing. Then after that, the cells from here come down and start fusing downwards. That's what's happening. But you see, like when they started coming up, you see this is a hole right here, open area right here. This open area, you see right here, they're separating two parts because there's a fusion happens right here in the middle, and then you see there's a top part right here. And then you see a bottom right here. This top part, this open space, we call them an anterior neuropore, or this is called rostral neuropore. And this is called caudal or posterior neuropore. Okay, and this is really important. All right, so that's what I was trying to explain to you. All right, so when they come together, all right, when these guys come together, it started making, like, look, it started coming together, right? This is called. It's going to start coming in this portion right here. This portion right, it comes out, right, when we started folding together, right? When the neural foldings are folding. And then this gets separates out. Now this gets separates out and it becomes something like this. And this is what's called your, let me write down here. Call them your, let's say, neural tube right here. Okay? This is what's called neural tube. So because when these guys fold, like right here, some of the cells nearby here, they get disintegrated, meaning this cell separates out on the edge of this fusions. And these cells, on the edge of the fusions, with the cells separates out or get differentiated into these cells right here. These cells right here, look, this dot, all this. This is called your Neurocrash cells. So it's really important that you know this, right? So Neurocrash cells are like, forming. So when they're like forming right here, like on fusing, some of the cells down from here, look, from here, this cell is separating out and they get differentiated into something called Neurocrash cells. Because these Neurocrash cells are very, very important, for giving rise to like adrenal medulla, right? They're important for like, secreting epinephrine or norepinephrine, right? Not only that, they also make the, if you know from GI system, like Meissner complex or Auerbach complex, right? They give rise to like pseudo-unipolar ganglion, right? And Schwann cells also. These are some of the derivations of Neurocrash cells, and we'll talk about that too, right? But now, when these cells, right, when they get separated out, what is happening here? Again, this is going to be my precaudal plate. You're going to see a cloacal membrane right here. When separated, you see structure like this, and this structure right here, this is called neural tube, and then on top of the cells here, we call them Neurocrash cells right here, and this is a neural tube, okay? And this neural tube is the one. This neural tube, that's what's going to give rise to, let me write it. Neural tube is the one that's going to give rise to formations of your central nervous systems, your spinal cord, right? Central nervous system, spinal cord. What else? It's going to give rise to other bunch of stuff too. right? Like for example, like, if I could my oligodendrocytes, like retina too. All right. So look, this is neural tube, and then on there's going to be the cells right here. The cells are going to be here, are called Neurocrash cells, right? And you don't want to forget this structure right here. This is called again Notochord right here. All right. This is my endoderm, right? And this is going to be my ectoderm right here. Now, see, again, let's just label quickly. This is going to be your ectoderm, right? And remember, these mesoderms, these mesoderm also go, and get differentiated into different types of like mesoderms. Like this first layer right here, if I quickly make this first one, the first layer they make, like, let me just make this like, and this is what's called your paraxial mesoderm. And then the middle layer right here, they call them intermediate mesoderms. And then the lateral part, this is called lateral mesoderm, all right? And this is obviously this endoderm, which is going to make your like foreguard, midguard, and hindguard, all that kind of stuff, right? Now, so quickly, let's review this. The notochord, okay, induces the formation of your neuroplate, right? That neuroplate, when they go folding, what is going to happen here? When they go folding, they're going to make, from the midline, they're going to make what? The two structure, which is called anterior neuropore, and also called the posterior neuropore, right? And then after that, what is happening? Once it fuse, this gets, because this come and they separate out and become a neural tube, all right? Now, the importance of this is that there's something that I have to talk about. Look guys, when this is closing, okay, this is the cephalic end, and this is a rostral end, right? Sorry, caudal end. So when this is called anterior neuropore, what do we call them? And let me draw this actually. So let me make this. Look, if I make this right here, right? Look, there's going to be, okay, you can think about this structure right here. This is going to be your anterior neuropore, right? And this is called this is going to be posterior, because this is probably going to be like, yeah, so this is going to be a posterior neuropore, and this is going to be your anterior neuropore. Let's just make this, okay? So look, if we say this like posterior, anterior, and what is happening down here is that, let's cross this out. What is happening here is that, you guys already know that, what is this cover? This is called amniotic cavity, right? Amniotic cavity. Amniotic cavity, right? So what is happening here is that, sorry, I was right about this. It's called anterior neuropore, and that's posterior neuropore, right? So, and then remember, this guy's right here, right? So, this anterior neuropore, they usually get closest around day 25. Anterior neuropore, they fuse together around like day 25, and then different books might say different things, but it's about 24 or 25 days. And the posterior neuropore fuses around at day 27. Okay, so there's importance of this. Clinically, it's really important, because sometimes what happens is that this anterior neuropore does not fuse. If the anterior neuropore does not fuse, what is going to happen? That's going to give you a condition called, because it is happening upper, it's going to give you something called upper, right? Let's say upper neural tube defects. Neural tube defects. And what is that called? We call them anencephaly, right? Cephaly means brain, right? So anencephaly. It's going to make anencephaly, which basically means that your brain, okay, your cranium, the cranium vault, that does not form, right? That doesn't form. And on top of that, what is going to happen is that, some of the fluid, look, inside the neural tube, you have to remember this, there's a lumen too, right? That lumen, that lumen, what is that lumen called? That lumen's, okay, because that lumen is later that's going to form your ventricular systems. That lumen contains like CSF fluid inside the neural tube, like inside the neural tube right here, there's a CSF fluid, right? And if this, let's say, neuropore does not close, then what happens is that this structure from the neural tube, some of the CSF fluid started like, spilling out. they split out to, let's say, from the anterior neuropore, split to out the amniotic cavity. And later they can also go to your maternal, right, to the mother. So if you check your mother's serum, you'll see increased level of, let me write them, high level alpha fetoprotein, and also increased level of acetylcholine esterase. You get to see that. Okay, so whenever there's a failure of closing your, let's say, anterior neuropore, you're going to see the increased level of alpha fetoproteins and ACHE, right? Acetylcholine esterase. That basically tells us that, somehow the anterior neuropore did not fuse at a time, which is around like day 24 and day 25. And then same thing with the posterior neuropore too. If the posterior neuropore does not fuse, then this posterior neuropore is the one that gives rise to our spinal cord, right? So if this posterior neuropore does not close, then you get a lower, let's say we can say lower, this is posterior, we can say lower neural tube defects. And what do we call them? We call them a spina bifida. There are different types of spina bifida, but as a complex of this condition, we call them spina bifida. Okay, so, and this usually happens is the posterior neuropore that closes around day 27. Now after we know this, what could be the reasons why this anterior neuropore or posterior neuropore does not fuse? And that has to do with the very, very important hormone, and that is called folic acid. It is very, very important that, mother have, good amount of folic acid supplement, because if there is a deficiency of this folic acid, then what that could lead into is that this diffusion of this anterior neuropore and posterior neuropore may not occur. So if that doesn't occur, the CSF fluid that's in the lumen, that can get spilled out in the amniotic cavity. And those CSF fluid are your alpha fetoprotein, also acetylcholine esterase, okay? And this is something clinically you need to remember this, all right? And whenever we talk about, let's say, lead conditions, we'll go more depth about this. But I have to mention about the importance of the folic acid, especially during the pregnancy, okay? Especially, and this is taking place in third week. This is why, okay, even in this, when you're developing a nervous system, the main organs are forming, like after a third week, like nervous systems and all. That's just why it's also like importance of alcohol. Also, you don't want to take it, okay? Because that's going to give it can have a lot of defect, a lot of problems that can occur also, all right? Now after this, after these conditions, now we have to talk about what is going to happen with, what is this? That's what we talk about, right? Now, so you see this structure right here? What I did was let's say I caught it more in like sagittal. I got a sagittal of this structure right here, okay? And I'm just showing you in one. So this is basically, look, this top right here, this top right here, you can simply say this is a lumen right here, okay? And then this guy is right here. This is going to be your, let's say, upper neural tube. And this is your lower neural tube. Or simply we can call them, this is a cephalic end and this is a caudal end. Because the cephalic, this one is the one that is going to form your brain, and this caudal is the one that's going to form your, like a spinal cord. All right. So now, if you see this structure right here, they started coming together, right? And then they started fusing, and they move upward and they close at the day 25. And then after that, they will start zipping downwards and they close it around day 27. So this is going to be your anterior neuropore, and this is going to posterior neuropore of this structure, which is your neural tube. Because what I did was that, you just cut the neural tube, okay, and then you show it more in this view. All right. So when you say it looks something like this, right? Now, and this is what? This is the neural tube structure. This is a neural tube structure that you're seeing right now. This is a neural tube structure right here. All right. Now, if I just, let's say, rather than making like difficult, if I just make a tube, let's say this is a neural tube right here, right? So you just make a tube. You can imagine this is a tube. What happens is that this neural tube started going morphogenesis. What does that mean? Which basically means that this started neural tube will go process called vesiculogenesis, meaning that this neural tube we started forming like swellings. What they started forming? They started forming swellings. Okay, and they, first what they do is they form like very, very important swellings. There are 3 of them they form right here. Look, this first one, we call them a pro, zen. Pro basically means what? Beginning, pro-encephalon. Encephalon means brain, right? This structure already is called pro-encephalon. And then, look, right here. This one right here, in the middle portion right here, this middle we call them a mesencephalon. And then this is called your rhombencephalon. All right? Remember, prosencephalons are also called forebrain. And the mesencephalons are called midbrain. And then these rhombencephalons are called your hindbrain or hindbrain. And then obviously you guys know this is a spinal cord. And remember, in this structure, okay, because the 3 swellings are formed, initially that's a 3 swelling because this neural tube started like, going through the process of vesiculogenesis, meaning that they started forming like vesicles, they started forming the swellings. And these swellings are called your prosencephalon, your mesencephalon, and your rhombencephalon. And obviously you see this spinal cord right here, okay? And you don't want to forget this. What is inside there is what? We call them up. This is what? This is a lumen right here, right? And look, from up to here, they're going to form, like, let's say, prosencephalon, mesencephalon, rhombencephalon. These guys, right, they will, make this lumen, they'll contain the CSF, right? So they start making the ventricular system, right? And then the spinal cord, they'll make a central canal, okay? This lumen. So you have to remember that too. Now, not only this happens, these guys even go further modifications. After forming the prosencephalon, mesencephalon, and rhombencephalon, some of this, like especially this prosencephalon and the rhombencephalon, they go further modifications. They get differentiated into 2 line structure, 2 more structures. Look, if you see right here, look, if you come here, look at this structure. The first structure right here, you see this right here? You guys know this structure, right? Because this is this structure, they get differentiated. One of them is called telencephalon, right? And then and these are rapidly growing cells, so they started forming the structure which is called, swelling called telencephalon. And this in black, it will form another structure which is called your diencephalon. And then this is what? This is going to be a mesencephalon. So mesencephalon stays the mesencephalon. It doesn't really go any changes. So let's just say this is a mesencephalon right here, all right? And then this is, guys, look, remember, it was rhombencephalon also go into further 2 modification. It creates a 2 swelling right here. And this one is going to be first, is going to make 2 swelling. One is called, let me write them. One is called metencephalon, all right? And the other one is called myelencephalon, all right? Remember, this metencephalon is the one that gives rise to pons and then your cerebellum, all right? Not cerebrum, cerebellum. And this, myelencephalon is going to give rise to your medulla oblongata. All right? So that's one. And remember, the telencephalon is the one that gives rise to what? Telencephalon gives rise to, like, your basal ganglia, your limbic system, all the lobes, like, for example, your parietal lobe, your frontal lobe, your occipital lobe, your temporal lobe, and also those insula lobe. Okay, it gives rise to that, basal ganglia. And this diencephalon is the one that's going to make what? That's going to make, down here, what is it? There's going to be if you make this thalamus right here, you can simply say this is my thalamus right here. Okay? And then, what is going to happen? They'll also make, I could just make this as let's just make this as what? Hypothalamus. It also makes hypothalamus. Soft thalamus is going to be there because if, let's say this is my hypothalamus, there's going to be soft thalamus. Okay, let's say the soft thalamus. They also make epithalamus, right? Also like mammillary bodies. All right, so this diencephalon is the one that's going to give rise to all the thalami, right? And then telencephalon is the one that's going to give rise to your cerebral cortex that contains what? Like as I said, like parietal lobe, occipital lobe, temporal lobe, insula, limbic system, like formation of the hippocampus or amygdala, that is actually coming from the telencephalon. Okay, and that is what? And that is a derivative of your prosencephalon, right? And who is giving rise to prosencephalon? Your neural tube, because that neural tube created the swelling, right? That's what's going to happen. And not only that, and the mesencephalon, that's going to be midbrain, right? And then, look, the last portion, which is rhombencephalon, which also going for the two swellings, it splits into two swellings. One is called metencephalon. And from the metencephalon, it gives rise to pons. And then from the posterior, look at this. If I make this like, what is this? This is your mind? That's a cerebral cortex down here, right? You can see that there's temporal, like parietal, frontal lobe here, and then occipital lobe here, right? So, and this is just like the sagittal view. And then this is my midbrain right here. You see this is right here? Behind, this is what? Behind this guy right here, let's say this is my pons. Behind the pons, because this is pons and medulla right here, right? Behind the posterior of the pons, there's going to be another swelling structures, okay? And that is what's going to form your cerebellum, right? And then obviously, the myelencephalon is the one that's going to give rise to your medulla oblongata, right? And this is, you guys know this is what? Spinal cord. All right? And this is your cerebellum, medulla, and the cerebral cortex, right? Now, this is how, the nervous system develops.